Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Nucleolus. 67 languages. Nucleolus contained within the cell nucleus. Cell biology. Animal cell diagram. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. 8 smooth endoplasmic reticulum. For the solution concept in cooperative game theory, see Nucleolus, game theory. The nucleolus, /nucleolus, nucleolus /pl. Nucleoli /la / is the largest structure in the nucleus of eukaryotic cells. 1. It is best known as the site of ribosome biogenesis. The nucleolus also participates in the formation of signal recognition particles and plays a role in the cell's response to stress. 2. Nucleoli are made of proteins, DNA and RNA, and form around specific chromosomal regions called nucleolar organizing regions. Malfunction of the Golgi apparatus means that nucleotide is the cause of several human conditions called nucleolopathies. 3, 4, and the nucleolus is being investigated as a target for cancer chemotherapy. 5, 6. History, edit. The nucleolus was identified by bright field microscopy during the 1830s. 7. Theodore Schwann in his 1939 treatise describes that Schleiden had identified small corpuscles in nuclei, and names the structure Kernköpfchen. In a 1947 translation of the work to English, the structure receives the name of nucleolus. 8, 9. In addition to these peculiarities of the cytoblast, already made known by Brown and Main, Schleiden has discovered in its interior a small corpuscle, see Plate I, Fig. 1. 4, which, in the fully developed cytoblast, looks like a thick ring, or a thick-walled hollow. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. 13 centrosome. 14 cell membrane. Globule. It appears, however, to present a different appearance in different cytoblasts. Sometimes only the external sharply defined circle of this ring can be distinguished, with a dark point in the center, occasionally, and indeed most frequently, only a sharply circumscribed spot. In other instances this spot is very small, and sometimes cannot be recognized at all. As it will frequently be necessary to speak of this body in the following treatise, I will for brevity's sake name it the nucleolus, Kernköpfchen. Nucleus corpuscle. Theodore Schwann, translated by Henry Smith, Microscopical Researches into the Accordance in the Structure and Growth of Animals and Plants, page 3. Little was known about the function of the nucleolus until 1964, when a study, 10, of nucleoli by John Gurdon and Donald Brown in the African clawed frog Xenopus levis generated increasing interest in the function and detailed structure of the nucleolus. They found that 25% of the frog eggs had no nucleolus, and that such eggs were not capable of life. Half of the eggs had one nucleolus and 25% had two. They concluded that the nucleolus had a function necessary for life. In 1966, Max L. Bernstein and collaborators showed via nucleic acid hybridization experiments that DNA within nucleoli codes for ribosomal RNA. 11, 12. Structure, edit. Three major components of the nucleolus are recognized: the fibrillar center, FC, the dense fibrillar component, DFC, and the granular component, GC. 1. Transcription of the rDNA occurs in the FC. 13. The DFC contains the protein fibrillarin, 13, which is important in RNA processing. The GC contains the protein nucleophosmin, 13, B23 in the external image, which is also involved in ribosome biogenesis. However, it has been proposed that this particular organization is only observed in higher eukaryotes and that it evolved from a bipartite organization with the transition from anamniotes to amniotes, reflecting the substantial increase in the DNA intergenic region. An original fibrillar component would have separated into the FC and the DFC. 14. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Nucleus from a cell line. Fibrillarin in red. Transcription. Regulatory protein CTCFL in green. Nuclear DNA in blue. Electron micrograph of part of a HeLa cell. The image is a screen capture from this movie, which shows a Z-stack of the cell. Another structure identified within many nucleoli, particularly in plants, is a clear area in the center of the structure referred to as a nucleolar vacuole. 15. Nucleoli of various plant species have been shown to have very high concentrations of iron, 16, in contrast to human and animal cell nucleoli. The nucleolus ultrastructure can be seen through an electron microscope, while the organization and dynamics can be studied through fluorescent protein tagging and fluorescent recovery after photobleaching, FRAP. Antibodies against the PAF49 protein can also be used as a marker for the nucleolus in immunofluorescence experiments. 17. Although usually only one or two nucleoli can be seen, a diploid human cell has 10 nucleolus organizer regions, NORs, and could have more nucleoli. Most often multiple NORs participate in each nucleolus. 18. Function and ribosome assembly, edit. Main article, Ribosome biogenesis. In ribosome biogenesis, 2 of the 3 eukaryotic RNA polymerases, Pol I and Pol III, are required, and these function in a coordinated manner. In an initial stage, the RNA genes are transcribed as a single unit within the nucleolus by RNA polymerase I. In order for this transcription to occur, several Pol I-associated factors and DNA-specific transacting factors are required. In yeast, the most important are UAF, upstream activating factor, TBP, Tatabox binding protein, and core binding factor, CBF, which bind promoter elements and form The potentiation complex, PIC, which is in turn recognized by RNA polymerase. In humans, a similar PIC is assembled with SL1, the promoter selectivity factor, composed of TBP and TBP-associated factors, or TAFs, transcription initiation factors, and UBF, upstream binding factor. RNA polymerase I transcribes most RNA transcripts, 28S, 18S, and 5.8S, but the 5S RNA subunit, component of the 60S ribosomal subunit, is transcribed by RNA polymerase III. 19. Transcription of RNA yields a long precursor molecule, 45S preRNA, which still contains the internal transcribed spacer, ITS, and external transcribed spacer, ETS. Further processing is needed to generate the 18S RNA, 5.8S, and 28S RNA molecules. In eukaryotes, the RNA modifying enzymes are brought to their respective recognition sites by interaction with guide RNAs. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF, which bind these specific sequences. These guide RNAs belong to the class of small nucleolar RNAs, SNRNAs, which are complexed with proteins and exist as small nucleolar ribonucleoproteins, SNRNPs. Once the RNA subunits are processed, they are ready to be assembled into larger ribosomal subunits. However, an additional RNA molecule, the 5S RNA, is also necessary. In yeast, the 5S rDNA sequence is localized in the intergenic spacer and is transcribed in the nucleolus by RNA polymerase. In higher eukaryotes and plants, the situation is more complex, for the 5S DNA sequence lies outside the NOR and is transcribed by RNA pol III in the nucleoplasm, after which it finds its way into the nucleolus to participate in the ribosome assembly. This assembly not only involves the rRNA, but also ribosomal proteins. The genes encoding these R proteins are transcribed by pol II in the nucleoplasm by a conventional pathway of protein synthesis, transcription, preRNA, processing, nuclear export of mature mRNA, and translation on cytoplasmic ribosomes. The mature R proteins are then imported into the nucleus and, finally, the nucleolus. Association and maturation of RNA and R proteins result in the formation of the 40S, small, and 60S, large, subunits of the complete ribosome. These are exported through the nuclear pore complexes to the cytoplasm, where they remain free or become associated with the endoplasmic reticulum, forming the rough endoplasmic reticulum, RER. 2021. In human endometrial cells, a network of nucleolar channels is sometimes formed. The origin and function of this network have not yet been clearly identified. 22. Sequestration of proteins, EDIT. In addition to its role in ribosomal biogenesis, the nucleolus is known to capture and immobilize proteins, a process known as nucleolar detention. Proteins that are detained in the nucleolus are unable to diffuse and to interact with their binding partners. Targets of this post-translational regulatory mechanism include VHL, PML, MAM2, POLD1, Relay, HAND1 and HTERT, among many others. It is now known that long non-coding RNAs originating from intergenic regions of the nucleolus are responsible for this phenomenon. 23. See also, edit. Differential interference contrast microscopy. References, edit. 1 A.B. Sullivan J.M., Pythe, Cridge A.G., Engelk D.R., Ganley R., June 2013. The nucleolus, a raft. Adrift in the nuclear sea or the keystone in nuclear structure. Biomolecular Concepts. 4, 327786. Do. 10.1515/BMC 2012-0043. PMC 510006. PMID 2543650. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 2 Olsen Mo, Dund M, the 16th of February 2015. Nucleolus, Structure and Function. Encyclopedia of Life. Sciences, Els. Do. 10.1024/9780470015902.0005975.pub3. ISBN 978-0-470-01617-6. 3 Hetman M., June 2014. Role of the nucleolus in human diseases. Preface. Biochimica it. Biophysica Acta. 1842, 6757 do. 10.1016/j.badies.2014.03.004. PMID 246316555. 4 Bahadari, M., Azizi, M.H., Dabayari, S., Bahadari, N., 2022. Effects of human nucleolus upon guest viral life, focusing in COVID-19 infection, a mini review. Iranian Journal of Pathology. 17/117. Do. 10.306994/IJP.2021.540305.2744. PMC 8794558. PMID 35096082. 5 Quinn J.E., Devlin Jr., Cameron D., Hanan Kilometer, Pearson R.B., Hanan Road, June 2014. Targeting the nucleolus for cancer intervention. Biochimica it Biophysica Acta, BBA, Molecular Basis of Disease. 1842, 680216 do. 10.1016/j.badies.2013.12.009 HDL. 11343/44176. PMID 2438939. 6 Woods S.J., Hanan Kilometer, Pearson R.B., Hanan Road, July 2015. The nucleolus as a fundamental regulator of the 53P response and a new target for cancer therapy. Biochimica it Biophysica Acta. BBA, Gene Regulatory Mechanisms. 1849, 78219 do. 10.1016/j.bagm.2014.10.007. PMID 2546432. 7 Pedersen T., March 2011. The Nucleolus. Cold Spring Harbor Perspectives in Biology. 3, 3. A000638. Do. 10.1101/kshperspect.a000638. PMC 3039934. PMID 21106648. 8 Mikroskopisch Untersuchungen über die Berinstimung in der Struktur und dem Wachstum der Tiere und Pflanzen / von th. Schwann. MIT via Kupfertafel. Wellcome Collection. Retrieved. The 12th of March, 2024. 9 Schwann, Theodore, Smith, Henry, Schleiden, M.J. 1847. Microscopical. Researches into the accordance in the structure and growth of animals and plants. London. Sydenham Society. p. 10 Brown D.D., Gurdon J.B., January 1964. Absence of ribosomal RNA synthesis in the A nucleolate. Mutant of Xenopus levis. Proceedings of the National Academy of Sciences of the United States of America. 51, 113946. Bibcode, 1964 NAS, 51139b. Do. 10.1073/NAS.51.1.139. PMC 300879. PMID 1410667 11 Bernsteel ML, Wallace H., Serlin J.L., Fishberg M., December 1966. Localization of the ribosomal DNA. Complements in the nucleolar organizer region of Xenopus levis. National Cancer Institute. Monograph. 23, 43147. PMID 5963987. 12 Wallace H., Bernsteel ML, February 1966. Ribosomal cisterns and the nucleolar organizer. Biochimica et Biophysica Acta, BBA, Nucleic Acids and Protein Synthesis. 114, 2296310. Do. 10.1016/0005-2787, 66, 90311X. PMID 5943882. 13 ABC Sarivi, Urquki Inchima S., Russell P., Hernandez Verdun D., January 2008. Nucleolus, the fascinating nuclear body. Histochemistry and Cell Biology. 129, 11331 do. 10.1000/S00418-007. 0359-6. PMC 213797. PMID 1804651. 14 Thierry M., Lafontaine D.L., April 2005. Birth of a Nucleolus, the evolution of nucleolar compartments. Trends in Cell Biology. 15, 41949 do. 10.1016/J.TCB.2005.02.007. PMID 1581735. As PDF. Archived 17 December 2008 at the Wayback Machine. 15 Bevanaf, Leah, Razas M., Leader DJ, Brown J.W., Shaw P.J., June 1996. The organization of ribosomal RNA processing correlates with the distribution of Nucleolus NAS. Journal of Cell Science. 109, pt 6, 1241-51 do. 10.1242/JCS.109.6.1241. PMID 8799814. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 16-carat Roskstets H, Grillit L, Ezor MP, Conjuro G, Ortega R, Curie C, Marie S, August 2011. Plant cell Nucleolus as a hotspot for iron. The Journal of Biological Chemistry. 286, 32278636. Do 10.1074/jbc.c111.269720. PMC 3151030. PMID 2171970. 17-carat PAF49 antibody GeneTex Inc. Genitex.com. Retrieved 18 July 2019. 18-carat von Nebel-Doberitz M. Wensensen N. 2008. The Cell, Basic Structure and Function. Comprehensive Cytopathology, 3rd ed. 19-carat Champ PC, Harvey Ra, Ferrier Dr. 2005. Lippincott Illustrated Reviews, Biochemistry. Lippincott Williams and Wilkins. ISBN 978-0-7817-2265-0. 20-carat Alberts B., Johnson O., Lewis J., Raff M., Roberts K., Walter P., 2002. Molecular Biology of the Cell. 4th ed. New York, Garland Science. pp. 3313. ISBN 978-0-8153-3218-3. 21-carat Cooper G.M., Horsman Re., 2007. The Cell, A Molecular Approach, 4th ed. See Nauer Associates. pp. 3719. ISBN 978-0-87893-220-7. 22-carat Wang T., Schneider J., 1 July 1992. Origin and Fate of the Nucleolar Channel System of Normal Human Endometrium. Cell Research. 2, 297-102 do. 10.1038/CR.1992.10. 23-carat Audaste, Jacob M.D., Lee S., January 2012. Immobilization of proteins in the nucleolus by ribosomal intergenic spacer non-coding RNA. Molecular Cell. 45, 214757. Do 10.1016/J.Molchel.2011.12.012. PMID 2228465. Further reading, edit. Cooper G.M., 2000. The Nucleolus. The Cell, A Molecular Approach, 2nd ed. Sunderland Ma. See Nauer Associates. ISBN 978-0-87893-106-4. Tiku V. Antebia, August 2018. Nucleolar Function in Lifespan Regulation. Trends in Cell Biology. 28, 8. 662672. Do 10.1016/J.TCB.2018.03.007. PMID 297798666. S2 SID 2916718. Joanna Klein, 20th May 2018. The thing inside your cells that might determine how long you live. The New York Times. External links, edit. Nucleolus under electron microscope 2 at Unimains. Nuclear protein database search under compartment. Cell and nucleolus at the U.S. National Library of Medicine. Medical subject headings, mesh. Histology image, 2010. Follow a histology learning system at Boston University. Categories, organelles, nuclear substructures. Explore our developer-friendly HTML to PDF API printed using PDF Crowd. HTML to PDF.